Good morning, children. Let us continue with the lesson photosynthesis, and today we are going to study about immersion's effect and involvement of two pigment system. The most effective light for photosynthesis is red light, 650 to 680 nanometers, and photosynthetic efficiency decreases in far red light. That is, you've studied about Vibgyor, the spectrum, and when we move away from what happens from red to far red light, photosynthetic efficiency decreases. Now in 1950s, Robert Emerson and his associate observed that if light of a shorter wavelength was superimposed by light of far red wavelength, rate of photosynthesis was greater than could be expected from adding the rates with either light when given alone. Now, this enhancement or increase in rate of photosynthesis in far red light by a light of shorter wavelength is said to be immersion enhancement effect. So what has happened here? That rate of photosynthesis in far red light was lesser, and when it was superimposed by a light of shorter wavelength, the rate of photosynthesis increased, and this is called immersion enhancement effect. Then D.J. Peary and M. Gibbs, they studied the rate of photosynthesis in isolated spinach chloroplasts. Now what they did was that they irradiated the chloroplasts with lights of wavelength 640 and 720 nm. That is a light of shorter wavelength and a light of a longer wavelength. So what they did was they irradiated the chloroplasts with lights of wavelength 640 and 720 nm. When the two lights were supplied simultaneously, one after the other, the rate of photosynthesis was about 25% higher than the sum of rate of photosynthesis in the two lights separately. Like if first you gave 640 nm and you noted down the rate of photosynthesis, then you gave the light of 720 nm and you noted down the rate of photosynthesis, and you added the increase in both, then also what happened that the two lights were supplied simultaneously, rate of photosynthesis was about 25% higher. Okay, thus both lights had a synergistic effect on photosynthesis. That is, one helps the other in carrying out the process. It leads to the conclusion that two groups of pigments, one pigment absorbing lower range and the other higher range of wavelength, are involved in photosynthesis. These pigments are organized into two distinct or discrete photochemical light harvesting complexes within the photosystem 1 PS1 and photosystem 2 PS2. These are named in the sequence of the discovery and not in the sequence in which they function during the light reaction. Okay, so in our previous class, we had studied that the light harvesting system, it is made up of hundreds of pigment molecules, and they are bound to proteins. And each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. Single chlorophyll A molecule forms the reaction center. We did it in our previous class. And in photosystem 1, the reaction center chlorophyll A has an absorption peak at 700 nm, whereas in photosystem 2, it has the absorption maximum at 680 nm. So, two photosystems are involved, photosystem 1 and 2. Photosystem 1 includes pigment which maximally absorb light of wavelength longer than 680 nm. You have to remember that. Photosystem 1, what is it doing? It is absorbing light of wavelengths longer than 680 nm. and this system is designated as P700, and they are driven by light of shorter wavelengths as well. Photosystem 2 includes pigments which absorb light at 680 nm and lower wavelength and is designated as P680. Chlorophyll A is the key pigment in both photosystem 1 and photosystem 2, and they correspond to two reaction centers of energy trap where light energy is converted into chemical energy. The pigments which constitute the photosystems make only about 1% of the total cellular pigments. The number of photosynthetic pigment molecules necessary to effect a photochemical act constitute photosynthetic unit. Now, both photosystem, they act in a series to produce NADPH and ATP, which are the end products of light reaction of photosynthesis. In this process, electrons flow from water and they reduce NADP to NADPH, and the energy needed to remove electrons is provided by light quanta absorbed. So the pathway of electron transport from water to NADP involves two photosystems and is often called the Z scheme because it looks like the alphabet Z. So this was the Peary and Gibbs experiment where he discovered two types of photosystems acting together, and in the case of spinach chloroplasts. Photophosphorylation. The process through which ATP is synthesized by the cells is called photophosphorylation. Photophosphorylation is synthesis of ATP from ADP, that is adenosine triphosphate from ADP and inorganic phosphate in the presence of light. So, photolight phosphorylation is the synthesis of ATP from ADP. Now, photophosphorylation is accomplished through two cycles, cyclic and non-cyclic electron transport system. Now, what happens when the two photosystems work in a series? The first photosystem 2 and then photosystem 1, then you call it non-cyclic photophosphorylation. And on the other hand, when only photosystem 1 is functional, the electron is circulated within the photosystem and photophosphorylation occurs due to cyclic flow of electrons. So what is the difference here? That when two photosystems are working in a series, you call it non-cyclic, and only when one photosystem is functional or photosystem 1 is functional, you call it cyclic photophosphorylation. So now we begin with non-cyclic photophosphorylation. You can see that, as I told you earlier, there are two photosystems working in unison, and non-cyclic photophosphorylation is the main process in which plants harvest the light energy. It is accomplished through non-cyclic ETS or electron transport. And here you can see in the diagram, both photosystem 1 and photosystem 2 are involved. Now you have studied in your previous class, you can see that the light energy is absorbed by the two photosystems, and then you can see through arrows that there is the reaction center from where an electron has been expelled. The electron expelled by the reaction center pigment molecules, they are not returned back to them. Okay, so that is why it becomes non-cyclic. You can see that the electron that has been released is being accepted by a primary acceptor, then it by ferredoxin, and then you can see the electron is transported to NADP. Okay. Now here what happens, that in this system you can see photosystem 1 and 2, and in this photosystem 2 you can see that photolysis of water has occurred and electrons have been 4 molecules of this water hydrogen have been released that are used to reduce NADP to NADPH. So you have reduced NADPH that is further utilized in the dark reaction of photosynthesis. So oxidized P680 returns to its ground state by regaining the electrons released by photolysis of water. Oxidized P700 comes back to its ground state by gaining electrons donated by oxidized PC associated with photosystem 2. So you can see the various steps here, and non-cyclic photophosphorylation needs a constant supply of water molecules to be oxidized and NADP to be reduced. As you can see here in photosystem 2, there is a continuous supply of water, that is the main necessity for water molecules to be oxidized and NADP to be reduced. Now we come to cyclic photophosphorylation. Here, as I told you earlier, it involves only one photosystem and the site is stroma lamellae. While the membrane or lamellae have both photosystem and two stroma lamellae membranes, they lack PS2 as well as NADP reductase enzyme. So here photosystem 2 is absent and NADP reductase enzyme is also absent. The electron expelled by the reaction center P700 does not pass on to NADP but return back to photosystem after passing through a series of electron carriers. You can see the electron carriers are the ferredoxin, the cytochrome system, plastocyanin. Various steps in cyclic photophosphorylation can be seen here, and hence cyclic flow results only in the synthesis of ATP but not of NADPH or reduced NADP. Cyclic electrons flow and cyclic photophosphorylation occurs when the plant cell contains plenty of NADPH but requires additional ATP for other metabolic needs. Now let us see the difference in both of them. You have seen both of them. So now let's see the difference in non-cyclic and cyclic photophosphorylation. Non-cyclic, as you knew, it involves both photosystems PS1 and PS2, and cyclic involves only photosystem 1. Non-cyclic, ATP and NADPH are formed. But just now in cyclic you saw that only ATP is formed. In non-cyclic photophosphorylation, if you go through the diagram again, you will see that oxygen is liberated as a result of photolysis of water. And in cyclic, oxygen is not liberated. Then in non-cyclic, electron expelled by reaction center pigment molecule is not returned back to it. But in the case of cyclic, the electron expelled returns back to it. Then in non-cyclic, water is consumed in the process, and in cyclic photophosphorylation, water is not consumed in the process. So these are the five very important differences between the non-cyclic and cyclic photophosphorylation. In our next class, we will continue with the chapter. Thank you, children.